Hello, everybody. Welcome to the Decoding Soil Biology webinar that Biome is well promoting. Um, Today we like to talk about the WeRoot project in soil health. This is a project together with Biomakers and involve Fields Forever researchers in this specific topic. My name is Alberto Acedo, and I'm the co-founder and the chief scientific officer in Biomakers. My role in the company is to develop technology and try to decoding biological interactions to use microbiome as an indicator of different status of the soil health. Together with me, we have two top researchers in the world that are focused, in this case, in different aspects of the wheat. For me, it's a pleasure to present Maria Hernandez-Soriano and also Silvio Salvi. Please, Maria and Silvio, if you want to say more about you, go ahead.
Thank you, Alberto. Thank you so much for the introduction. Well, like he has said, I'm Maria Hernandez. I'm a postdoctoral researcher at UNINESCENT UK. And well, we have had now a long collaboration with Biomakers. We started to work together in 2020 thanks to the Fields Forever initiative. And recently we have been lucky to also be partners in a new funding project together with Silvio and some other partners we will introduce later.
Yes, thank you, Alberto. Thank you, Maria. My name is Silvio Salvi. I'm a full professor at the University of Bologna in agricultural genetics. And we deal with the genetics of many traits of agronomical importance, especially root traits. And so we try to map and clone genes important for this trait. And we consider the interaction between root traits and the microbes in the soil as a root trait. So we try to deploy the genetics tools in trying to understand more about this interaction. Thank you.
Thank you very much. Only a brief introduction about why today we are talking about wheat, about a project that combines microbiome with genetics and environmental features in different parts of the world. Well, today we are living different challenges at global level. The global population is growing, as always, but we are in a specific environment at political level, at environmental level also, the climate change, that allow us to go deep in be more efficient in the way that we can grow food, in this case, wheat, that is supporting many food diversity in the world, from bread to feed animals that is part of our nutritional part. And we need to be very efficient on that, and looking for new data that allow us to take this kind of decisions: what kind of genetics, what kind of management, what kind of stimulation we can run in the soil to get more yield, but also more quality food. About Biomakers, we started in 2015 in San Francisco, US. The two founders are from Spain, but now the company is a global company that involves more than 70 members in different locations of the world. We are working with more than 2,000 customers that today are using soil microbial interactions and indicators to promote the soil health and also some characteristics to achieve sustainability at farming level. We are actively in different parts of the world, receiving samples, decoding that interaction, and providing useful information to farmers, agronomists, retailers, to try to involve the best decisions possible. The company is basically a tech company with 22% of the people PhD in different levels, that in the last 7 years, we identified more than 10 different millions of microbes and collecting a lot of different samples in different crops and different countries that allow us to use the most or the bigger database in the world about soil microbiome. With that, we developed different applications to test the soil health status, for example, sustainability assessment or testing products on the field under a specific methodology, and also invite other companies to be involved in this specific big crop standard involving different licenses and integration of the technology, because we are providing a layer of information, the soil biology layer, that can be implemented in different platforms. About the initiative that we are doing, because soil biology is very complex, we try to partner with different researchers in the world in order to understand what can be the possibilities to use soil microbiome as discover different indicators. Fields Forever is the non-profit initiative that we launched 2 years ago that involved more than 180 projects in 40 different countries, and today we collected around 21,000 soil samples worldwide. Also, we have another action that involves the company per se, that we are reinvest 2% of our revenues in soil health actions. And for the last one, I invite all of you to enter in the Big Crop Advisor community, where we are changing and exchange different learnings about the soil health indicators that we are using in Big Crop. About the initiatives, the Fields Forever for me is the most interesting initiative. And today we are talking about one of the projects that involve the 180 different projects. That projects are classified in different 7 topics. And in this case, we are in the topic of soil health, sustainability, yield, and nutrition. The project that we can start, in this case, Maria is starting to talk, involve wheat as a crop, and also very important topic, how nitrogen can be used in a more efficient way growing this specific crop. Maria, it's your part.
Excellent. Let me share my screen now. I can see, how can I share my screen? I've lost the show my screen. There you go. That's so where people behind the scenes helping us screen monitor one. There you go. So hopefully you can see my screen now. I will hope that's the case. So yeah, we were. I want to say first that I'm a soil. Initially, I'm a soil scientist. I was a soil chemist during my PhD and most of my postdoc with a great interest in carbon. But when I joined Ines, I started to explore nitrogen, because obviously it's a very urgent question to solve, how we can make a better use of the nitrogen fertilizers for our soils to improve the sustainability of wheat crop production. Wheat provides 20% of the energy that we have every day and some of other important nutrients, and it's very much jeopardized by global change, conflicts, and the loss of soil. So that's how my interest, but obviously you cannot study the soil without the plant, or the plant without the soil. I think the interaction through the rhizosphere is key to improve any agricultural strategy. And well, I'm going to introduce you how when we became to be part of one of these 20,000, some of these 20,000 different samples of the Fields Forever, and how I could link this microbiome analysis that Big Crop, that Biome Makers run, with my interest in identifying the capability of wheat to control nitrification. So let me present. So it all starts with, yeah, like I was saying, 50% of the nitrogen fertilizer that we apply to our soils gets lost to the environment. It's quickly transformed by microbes in the rhizosphere, first to nitrate, which is still very good for the plant. It's easy to uptake, but it's very soluble, so it can be easily lost to the environment by leaching. And then it can be further transformed into nitrate, which is a very potent greenhouse gas. Synthetic inhibitors, inefficient, expensive, potentially contaminant. However, in recent years, as you can see in the slide, several plants, several species of several plants have been reported to have to release compounds in the rhizosphere that decrease this nitrification in a natural way. It's natural compounds, and even more recently, wheat has also been reported to have this capability, and that has implications. So when it's released through the rhizosphere, it can slow down this nitrification. And more recently, we are starting to actually report that it can also affect the nitrification. Now, this was first reported in just a culture of nitrosomonas in the laboratory, but we wanted to explore it in the field. We wanted to see if that impact was also happening in the, that control of the nitrification was actually happening in field conditions, which is ultimately what we are interested in. So we have a collection of wheat of more than 1,000 cultivars at UNINES. It's called the Watkins collection. It goes back 100 years, and we have land races that predate modern breeding. So there is a pool of traits that might have been lost or we don't have in modern cultivars that we can try to rescue from these historic land races and take just a bit of the genome that's of interest, and by natural breeding just introduce them into the modern varieties. So what we did is first, we explored the Watkins collection. We took a core collection of we took around 80 of the main representatives of the genetic variability of wheat variety variability across continents, and we grow them in the field. And what we confirmed first is that this elite variety has a very low BMI, and the Persia, which is this land race that has a high capability to suppress nitrification. When we examine the microbiome in the rhizosphere, you can see at this is at order level, you can see a nice segregation in the composition of the communities. So that it already confirms that there is indeed these exudates from the plant are having an impact on the composition of the community. Currently, our capability to predict functions from 16 analysis has gone a long way, and using PyGrass, we can also see there are differences in the actual functions that these nitrifying communities perform. So we are not looking just at one single bacteria, but any microbes in the soil that can actually have a role in the cycling of the nitrogen. And indeed, we confirmed, for instance, for ammonium monoxygenate, which is the example I have brought here, a significant reduction in this enzyme activity, in this transformation from NH4 to NO3 to nitrate. And then, obviously, once we have confirmed this effect from this land race, we want to see where in the genome of the wheat this trait is present. And nowadays, we can do that because the wheat genome has, in recent years, it has been fully sequenced. So something we do is to cross the low BNI, the low nitrification capacity variety, is a modern variety, with the one that presents this capability, and we obtain 87 different crosses. So each cross will have a bit of the modern one and a bit of the land race one. And then we grow all the 87 varieties in the field. We collect the rhizosphere soil, and well, Biome Makers had all the analysis done for us and sent us the results. And we found indeed that the differences in the different functions that we can predict with PyGrass and how it varies across the 87 individuals within the population. And then we do QTL analysis with this quantitative trait loci, which allows us, is, so to speak, a correlation between the phenotype effect, what these changes in the functions of the microbes with the regions of the genome. And we found both changes in the nitrifying communities linked to some bits of the wheat genome, but we also found bits linked to the actual function, the actual nitrogen cycling function of the microbes within the wheat genome. So that was in 2020. Obviously, to confirm this trait in field conditions, we need at least a second repetition. So last year, we put all the plants again in the soil, all the 87 triplicate and all the design perfectly done, and we collected the rhizosphere soil again. And just a couple of weeks ago, we have completed all the, I think it's like 300 analysis of the rhizosphere soil, and we are very much in the process of very soon confirm this trait. These preliminary results I presented in a conference a year ago, and then, well, it gave the seed to an idea to look for more beneficial root traits, because obviously introducing this capability for the plant for wheat to control nitrification has important implications: economic, to save money to the farmers in the nitrogen fertilizer; environmental, to reduce emissions; and obviously to continue producing the food, so obviously social. And that's when the Wish Roots was born, with these preliminary results and other root traits brought by our colleagues in the University of Bologna, which are more morphological traits that can also have an impact in soil structure, aeration, water retention capacity, which are morphological traits, and our colleague Silvio will cover in a few minutes. So the Wish Roots was conceived to bring these traits that are somehow hidden or lost in historical cultivars and bring it into modern varieties. So improving soil health is a bit our breeding target. The Wish Roots, as I'm going to present you in a minute, it's 7 different partners across 7 different countries, and it was funded in December through the European Joint Programme in Soil, and by 5 different funding bodies across the 6 different countries. There are a few faces missing here still, but as you can see, we have a large community so far. We have brought together soil scientists, microbial ecologists, and of course plant geneticists. And this is a really multidisciplinary effort to advance the sustainability of wheat production. Our main objective is tuning the wheat root microbiome to improve soil health, but also through the architectural traits, and optimize nitrogen cycling and availability, so the plants to improve nitrogen use efficiency, of course. There is a bit more in detail our objectives. So basically traits related to soil structure and nitrogen cycling. And once we can phenotype these traits, identify them in which cultivars or in which even bits of the genome they are present. And once we have this information, we can provide it to breeders to introduce these beneficial traits into modern elite varieties and advance the production. So here is just a kind of visual summary of these architectural traits we are seeking, so we are trying to identify, and of course the functional ones that are linked to the exudate. So the architectural one, direct impact on water storage, aggregation, nutrient cycling. The functional ones, the release of exudates that can tune the microbiome, not just the composition of the communities, but the actual functions, which is obviously more important. And of course, this has the potential of introducing these traits that can improve soil health, and soil health also not just the qualities of the soil, but soil health also as the capability of providing a good substrate and all the nutrients necessary to grow our crops. As environmental, it addresses environmental, social, and economical sustainable goals. Again, we are in this now. We have for this project, we are not only going to use the bread wheat collection at Joannines. We have also access now through the University of Bologna to the durum wheat germplasm, which is nicely managed there. So we have a really nice pool of resources to find these traits that we are trying to identify. It has obviously having a multidisciplinary team, what it means that we are going to have a multidisciplinary approach. We are going to combine genetics, plant genetics, soil biology, physics, and soil chemistry. So that not only brings the multidisciplinary expertise, it also brings multidisciplinary methodological approach. So we are in a unique position to do the phenotyping from very different aspects and have a much more holistic approach. And then not only stop at the soil, but actually see how the plant and the soil are interacting and how we can make that interaction, optimize it for a better production of wheat. So we just started in April. That's all very recent events. Our first pilot trial started in April. We are approaching the harvest, but through 2023 and 2024, the same 20 varieties, 10 bread wheat and 10 durum wheat, will be grown in 6 different countries, as highlighted in the map, for 2 consecutive years, following a strategy. Obviously, we will explain in detail in our website or in other events, to have all the information about these traits, how they behave or how the plant behaves across all the different countries. So in 3 years' time, we'll have a lot of more information about these traits across different countries. All of this will be also supported by semi-field phenotyping, so that means we can support the field work with a more controlled environment, which not only will allow us to confirm this and more controlled approach, but also to expand the phenotyping to match more plants that will be run in Germany. And that's it for the moment. My colleague Silvio is going to now introduce the other house, not just the microbiome bit, and I have focused more. He's going to talk also about the morphological traits we want to identify. And after Silvio covers this aspect, Alberto will expand a bit more how Biome Makers is going to collaborate with us in the performing the microbiome analysis, but more importantly, how we are going to analyze and make the most of the data that we are going to obtain. Thank you so much.
So, I believe now you see my screen. Yes, here I am. So this is carry on with what Maria and Alberto presented, and I just wanted to let you know what we are basically contributing to the project of wish roots. So we are plant geneticists focusing on root genetics, and when you look at roots, you might want to look at roots in many different ways. And so in a genetics jargon, we call it traits and the component of the trait, and so on and so forth. So many root traits are actually very interesting for crops, and most of these traits you will see are very important also for soil health. One of the most important traits that we are working on is actually the rate between the resources that a plant is devoting to the root versus the resources that the plant is devoting to the shoot. So some sort of harvest index rate considering the root. Another very important trait is very, very much affected by root architecture is the anchorage to the soil, and also contributing to the resistance to root lodging or to lodging in general. Then we have a very large list of root spatial architecture traits, like the number of roots, the lateral roots originating from the axial roots, the root angle growth, the root distribution in the soil profile, but not only those traits which are kind of very, very trivial, but also the speed of growth of the root, the circummutation, so the way the roots are growing into the soil, which is never a very linear way, but somehow is some sort of helicoidal way. And so those roots that can be classified in the so-called tropisms of the roots. These traits also affect a lot the response to compact soil, to the soil structure more in general. We don't want to forget also the way that the roots, the participation of the root in plant-to-plant interaction, which is basically through root contacts into the soil. Then when we dig deeper into this type of morphological architecture type of standpoint, we also have to consider anatomical traits, and which is a kind of discipline at the very beginning. Not many people so far really investigated the quantitative variation for anatomical traits in roots across collection of germplasm, including wheat. So there is a large room for making scientific discovery in this area. Then we have a strong genetic control of the presence of root hairs. We have root exudations and the genetic control of those root and also carbon allocation to the soil. We already said that the importance of the genetic control from the plant side of the rhizospheric community of the microbiome growing around the roots, and all these traits are actually affecting the most important function of the root system, which is the nutrient and water absorption. How to phenotype the roots? So the roots can be phenotyped in many different ways nowadays, and I show you here a kind of quick excursus of the way that the roots can be investigated. We start from the very simple approaches like growing plants only for most of these examples are actually coming from wheat, which is the target species, one of the two target species in our study. And you start from a very simple system of phenotyping based on the investigation at the seedling stage of development, where you can grow a seedling either in very semi-hydroponic system with basically papers or wet papers, and or in soil in transparent pots. Then you can collect the phenotype by using appropriate image system and software system for root analysis. Or you can scale up a little bit and grow the plants in some sort of rhizoboxes or rhizotrons. This is the way that we call this structure, which are somehow kind of simple one, using a black perspex wall and a second glass wall in order to investigate in real time in a non-destructive way the way that the roots are developing. And again, you can digital picture at any stage and then analyze the data using appropriate software means. Or you can actually excavate the roots in the field, of course, with all the pros, all the plus and the minus of this approach. This is somehow called in the jargon of the field shovolomics, which makes a little bit funny the approach. And again, you can wash the roots and then collect it at any stage in this case, and then you can analyze the roots using appropriate software. Talking about the field, you just don't want always to work by hand, but you can also work by using machines. In this case, machines can be used by making tractor-aided machine using soil coring system. It can go deep in the soil level, extract portion of the soils, and then analyzing portion of them by looking for roots, which is a kind of mixture of automatized and manual approaches.
Sorry, because this is a super interesting part for me. Was incredible how to Silvio is able to see at the roots. And one of the questions that we have, I don't know if Silvio can try to solve something. If not, we can move ahead. But one of the questions that you are only focused on 16S. No, we are all focusing 16S and ITS to try also to identify fungi interested in relation with the root microbiome. But in the case of the fungi, it's more difficult to try to link taxa with specific functions because of the public databases linked with the genes or genetic potential of the fungi. That's a bit more complex than the bacteria. But yes, we are focused to both. Okay, I don't know what can happen with Silvio, but I think that we can move ahead with the presentation. Silvio, Maria, can you continue, Maria, or do you know a bit about the part of the Silvio that you can explain? Because in my case, for me, it's not possible.
I don't know, perhaps.
Okay.
I think he's still here, so perhaps I just
wanted.
Yeah,
he's here, but something happened with the sound. I don't know.
Yeah, I just wanted to add in something very interesting you just mentioned, actually, because obviously you can only present as much. When we first received the first results of this population of crosses between the two parentals with the contrasting nitrification inhibition. The first look at the diversity, we found even more diversity for the fungi, for the ITS that we found for the microbial communities. But then when we link it to the genome of the wheat, it was a strong QTLs, like really very strong significance with bacterial communities, not just the nitrifying, which was obviously our focus, but also many other interesting communities that we'd love to explore in the future. But no correlation between the wheat genome and the fungi. Nothing really striking. Nothing could be associated to the genome. So that's interesting how the different interactions, plant root soil behave. So I could try to continue a bit, just cover very quickly, because obviously I don't have Silvio's expertise. Definitely he and his team bring not only the germoplasm, bring years of expertise. They have identified, first thing I can say, so I'm showing my screen now. I think this is where Silvio left it.
Yes, that is possible, yeah.
Well, I know a bit. Obviously, I have come to many of his presentations, lucky me. And the one message that is very clear, since we are obviously running out of our time, is the power of imaging. Nowadays, we have the machinery, we can do shovelomics with a shovel, but we can also have all sorts of machinery to extract intact cores. So the times of field phenotyping being a dream, they are becoming to an end. I think field phenotyping is becoming more and more possible. But it's also how much imaging techniques have advanced. What just a simple photography of a nice photography of a root can tell us, it can tell us 10, 20 different morphological parameters that we can totally link with the wheat genome. And you can, what he was showcasing here is some reconstruction of how the wheat grows. I always like to say this, I'm passionate about the roots. Roots are having many million years in the plant. They know how to adapt to everything. So when there is a drought, they know where to find water. When there is poor nutrients, they know how to find their way. And if the soil gets compact, they will try to keep digging. So the plasticity of the roots is fantastic. And there is so many traits we can explore to improve crop production and also to make the soil better. So here is some of the work they have been doing that is now totally, you can look at all the papers they have published. It's different ways of phenotyping. So sometimes we do field phenotyping, sometimes we look very controlled situation. And these are different Coloseo and Lloyd, they are different durum weights. And what they are highlighting here is some of the different, how the angle of the root is very important. It will determine how the plant anchors, how it holds the soil. So it's good for the plant, it's good for the soil. And differences in this adaptation can have a crucial role in how the plant grows. So this is related to the angle of the crown in the soil. And there was also some, this is also it's a very nice story. This is some of the preliminary data that they were showing when I first met them. It's also about basically it's different morphological traits. Again, something about the angle, the length, depth, and how the root, the development of lateral hairs and things like that. All of that will allow the plant to be in the soil, but also to make the most of that in terms of nutrients, water, and obviously also linked to the microbiome in the sense that's how much they can reach to the microbes and control them in a way, or to tune a bit to the beneficial functions that they want. This is a bit similar, but obviously much more advanced because they have been working on this for a longer time. And so these are just regions of the durum wheat where they have found QTLs. That means regions that are significantly linked with this angle of the root, with many of the different morphological bits. Silvio, if you are listening to me, I hope I'm explaining it more or less understanding. And this is some of the recent, the recent research they have been published. And like I was saying, things like root hairs and root drivers. Obviously, it's all the roots are much more than anchorage and water absorption and nutrient absorption. They play so many different roles, and they also allow the plant to reach the microbes. And there is a very nice gradient, which is, I think, what Silvio was trying to highlight here. You could see that there are so many techniques we can see to see a gradient between when the roots exudates are released very near the root, the impact in the different microbial functions is incredible. I mean, you have such a shift in what the microbes are doing there because they are being very, very influenced by these root exudates. So hundreds of different compounds, enzymes, terpenes, hormones that are controlling the microbes. Obviously, this gradient fades as we go away from the root. Yes, some things we can measure obviously with the correlation with obviously that all is all connected. This is some of the beautiful imaging techniques for phenotyping that I was mentioning before. We have had so much advance in imaging, even techniques that were previously only used like medicine or for very specific. Now we can apply them, tomography, scanning, MRI scanning, to understand how the plant can adapt to grow under different circumstances and adapt to the challenges of our days. These are just examples of genes in the durum wheat that allow the plant to control elongation and grow more or less against gravitoprism in different conditions. I'm going to pass it quickly. Other mutants, this is a very, very recently accepted papers in PNAS, so I invite you to go and read it because Alberto, I suppose this material will be available later on in the website. So please feel free to contact Silvio. He's a fantastic teacher, so he's probably more than willing to explain a bit more in detail this finding. And of course, the links to the publications will be there. And with this overview, quick overview to the morphological traits, I think we can get back to the microbes in depth.
Okay, Maria, if you can help me with the presentation and you can continue putting your slides in the
okay, so
screen.
I can my face off.
No problem. I can continue talking, but when I can say next, you can go ahead.
I will be your.
Yeah. Thank you, Maria. Next, about microbiome. Yeah. About microbiome, it's not about the identification of the DNA chains that are present in the soil. There are different methods that we can use. For example, one of the typical questions is if we can identify dead or alive microbes, or in other words, if we can avoid extracellular DNA of the sample. That's something that we can implement from some specific trials that maybe are more interested to understand what are the specific cells that have the potential to grow, especially the more abundant one. Second is, okay, from the DNA, what are the information that we can get and we can use to try to correlate with environment, to try to correlate with root, with the genetics of the plant, everything. Typical works in microbiome are focused on taxa. That's something very easy. You have a specific DNA that you find and sequence, and from the DNA you try to match with public database to try to understand what kind of name you can assign, taxonomic name you can assign to that DNA. Sometimes it matches perfectly, sometimes there is a kind of variability, and also for that reason I mentioned before that in Biome Makers we have like a database of 10 millions of different DNA chains that most of them are not matching with the specific taxonomy at 100% that are published in database. That means that maybe we are identifying another species or another strains or another genera that is not described because only 1% of the microbes are able to be cultured. But one of the lessons working at globally is that in any part of the world that you take a sample, you can identify different group of taxonomic units. That's the same in animals or plants. If you are like measuring diversity in different jungles of the world. But for at the same that there is different taxons in different parts of the world adapted and evolved in different environments. Also, there are some functions that taxa is repeating all the time. For that reason, we need to go deep in the functionality of the microbiome. And also, another dimension that is very important, the ecology, because microbes can provide functions one per one, but that's not working like this. Microbes are working together, evolving together, and are in a specific community. Please, Maria, go ahead with the next slide. This is a representation of a microbiome community. This kind of representation, H dot, is the specific microbe that we find in a sample. The size of the dot represents the specific abundance. Can be relative abundance in percentage, or can be in quantitative abundance. We are using spikes like controls in order to try to quantify in absolute degree the number of the DNA chains that can be assigned to a taxonomic unit. And for the other hand, you can see some lines that are connecting one microbes with other microbes. Okay, lines are artificially. Okay, it's something that we are running in the computer, trying to calculate the two different things: the probability that two microbes are or we find that two microbes together in other samples, or the probability that we cannot find that two microbes in many times in a sample together. That allows us to understand ecological relationships in a basic way: positive and negative, co-occurrence that is positive, and co-exclusions that are negative. And also creating something that is named meta-community, that is the aggregating of different networks of the different samples, like to aggregate the different personal networks in Facebook or Twitter of each specific person that can be like a representative sample of a specific community or a city. And then we can start to measure some properties that are ecological properties. And Maria, can you click in the presentation? Or next? Yeah, perfect. That properties allow us to draw specific communities. No, for example, the number of clusters, the density of the network allow us to understand if there is more or less diversity, some kind of diversity in the sample. But also the number of connections. One microbe can be connected with only another microbe or with many microbes at the same time. That means that another properties like modularity, that in some ways or some communities allow us to see clusters. It's like there is an influencer of the community that is connected with other microbes, but that influencer is connected with another group of microbes that between them are not connected. Well, that properties are super interesting because we can explore specific functions that specific group of microbes that are creating a cluster are sharing. It's like, okay, this group of microbes are specialized in this kind of functions that come from the genome. The each microbe have a genetic potential, and we are exploring if that group of clusters or that cluster of microbes are sharing between them some kind of genes involved in specific metabolic pathways. That's how we are linking taxa, functions, and ecology in only one way. Please, Maria, next. Yeah, perfect. Well, looking at the clusters, for example, we can identify that one of the clusters is linked with some specific metabolic pathway, like nitrification or desnitrification. And because that clusters are in different density, in different abundance, and involve different pathways, we can try to get like a number that allow us to understand the balance of the different pathways, for example, linked with one nutrient, in this case, the nitrogen. This is a representation of the nitrogen cycle, and there you can see different genes that are involved in different pathways to transform some nitrogen chemicals into others. Depending on how bigger or no are that lines, we can understand that the cycle of the nitrogen potentially is in a direction that are releasing nitrogen or are allowing the plant to get nitrogen. That's how we are understanding the potential genetic of the microbiome to provide nutrient to the plant or not. Yes, next please. Well, this is my last slide. This is not linked with the results of this project, but I'd like to show you what can be the potential of this specific project with an example. This project that I present to you, or this data, is when we are taking data from environment, microbiome, genetics, different sources of data, we are applying one of our models that in this case is a supervised learning model, we are teaching the system with the specific samples that we know what can be the results, and we are testing the model with a few of that sample. In this case, it's a lasso with a regression model combined together. This data come from work from the University of Modena and Corteva in a project that is not in wheat, it's in corn, that also was involved in Fields Forever last year. In this case, we were testing not only one model, we ran different models. For example, only microbiome. What can happen if we try to understand the yield potential of a land only using the microbiome that is linked with a kind of corn variety or also environmental characteristics. In that case, we can get an accuracy that was in close to 91% using the data that we have in this project to test the accuracy. Well, we were testing the same only using the genetics. In this case, we involve only 4 different varieties, and only with the seed, with the genetics of the corn and other variables that involve physical chemical parameters and others, we get an accuracy to predict yield, or at least the potential to get more or less yield of 73%. Well, also we testing microbiome plus physicochemical properties or environmental properties, getting an accuracy of the 90%. That allows to get a conclusion. Microbiome is capturing some variables linked with the environment. For that reason, is describing the bibliography that microbiome can be a good indicator of a specific environmental properties. The last model that we ran is involving all kind of data for this project, microbiome plus physicochemical varieties plus feed genetics. And in this case, we get a super nice accurate on 98%. That means that in the samples that we were testing the result, in like 10% of the samples of the experiment, we get the right answer testing the model in this case specifically what can be the potential to get more or less yield. Next, please. Finally, with this kind of project today, in the case of the wheat, one of the things that of the potential impact that we can get is first, try to increase the yield that we are getting. How? Selecting the best specific genetics for the best specific environmental places, telling environmental biotic and abiotic factors. Biotic for the microbiome, what is the biological context, and abiotic, what can be the conditions that are linked with that place. In this case, also smart fertilization, if we understand what are the specific pathways that in certain conditions with specific genetics, what can be the best nitrogen fertilization possible, and also how to stimulate that pathways, that microbiome features, etc., to involve better nutrients and improve the quality of the food. For sure, there is a target of our mind that is promote soil health, promote biodiversity, promote the actions, or at least try to understand that the actions that we can run in the field can be supported the next generations. And for sure, the focus also in environmental impact. We are suffering losses on the wheat harvest this year in many countries, especially in South France, Spain, Italy, because environmental conditions, very drought conditions this year, etc., are supporting the numbers that we are losing, like more than between 30–40% of the yield compared with last year's, right? For that thing, this project I think is super interesting and allow to continue and support different aspects. Well, it's your turn. You were listening the last 30–40 minutes. It's time for receive your questions for me, Maria, or Silvio. Thank you very much. Well, there is a question from Michael that said, are you considering environmental factors, whether because 6 different countries have different agroecological zones? Maria, do you want to answer? Put your micro.
Classic. Yes, of course, we are going to be gathering, building on what Alberto was saying. Having accurate and broad metadata is very important to the success of this, to give some meaning to this microbiome analysis. So that means we are going to be gathering not only the soil, physical, chemical, biological, but we are going to link it to the different environmental factors, the soil types. We are going to do extensive characterization of the soil and obviously gather as much data about the environmental conditions, which might also change dramatically from one year to the other as we are, and from one country to the other as we are experiencing this year in many parts.
Yeah, we are running also different tests to understand how to tonificate, not only for weather conditions, also for biotic factors like microbiome. There is like a paper, for example, in Vineyard that we participate with the University of Copenhagen, that we describe how beta diversity is a super nice indicator of forms, or at least in the case of the vineyards, we are naming like terroir environmental features. But yes, it's not on. There are different ways to do that. We can consider that, because if we want that these results can be applied for the growers, one of the typical grower of any part of the world is, okay, this is my line, this is my region. This is very different to closer regions and far regions. All the results that you can get in 6 different locations with different environmental conditions can be applied to my field. For this reason, it's necessary to play with this modeling. There are 2 models to understand what are the features that can describe different characteristics that we are looking for, like yield, quality, or others. And also there are other models that try to predict, if you have a set of data from different parts and you control what kind of parameters you are looking for, or you can enter, then if you can take a sample for another place a bit far away, we can calculate with an estimation of error how the results that you learn from different places can be applied to that specific location. That's something that we are implementing in this project. For the other hand, we have another question, maybe it's for Silvio. I don't know, Maria, if you can answer, but if not, don't be worried, because all the questions that we are receiving, if we have no time, we can answer them specifically to you by email. They say, when you do the phenotyping of with roots architecture, why do you focus on early development stages? I would suggest to also do phenotyping after tillering and flowering and end formation. I don't know, Maria, if you plan to get root structure in different moments of the season, phenological states, etc., both at lab level and also in real conditions on the field.
Well, 2 parts. I think that's probably Silvio will have something to add. I think in my experience in the last years working in the world of root biology and root phenotyping, we had to make choices. There is the possibility, both in laboratory semi-field conditions, not so much in the field. In the field, there are obvious constraints about what Silvia was mentioning about shovelomics. So when you are, I have seen everything in terms of phenotyping in semi-field conditions, like you can grow an entire root system for up until maturity in laboratory conditions, in air or in soil or in solution. And then, of course, you have a control of how the root is growing, and you can totally reconstruct the entire architecture. If you pass me the question from the person, I can send lots of examples of that. Yes, sometimes we just are interested in early stages, and then we are also having constraint of the platforms we have. So it's very, when you want to grow plants in rhizoboxes, which are pretty much, I don't know, like an A4 sheet, You cannot let the plant grow for more than 2 weeks because then the information you're gonna get is not gonna be as good. So sometimes it depends on your research question. You can have enough information with a plant that has grown for 10 days. For other research questions, you want to excavate an entire system in the field that has been growing for 6 months. That obviously has a lot of technical difficulties because there is, for most of us, extracting just the crown for this kind of 15 centimeter is already as ambitious as we can go. Plus, after the flowering, that's something Silvio told me the other day, which I learned from him. The root is amazing in the field how quickly the roots will start degrading. There is really a quick kind of around the tillering or so on, between tillering and flowering, the plant is putting a lot of carbon in the soil. It has a lot of communication with the microbiome, and it has a lot of deposit of carbon. When it goes towards the flowering, it starts to be savvy, as I say. It's starting to get all the carbon from the photosynthesis, just keeping it for grain and for the rest of and the roots will start pretty much grading. So you don't learn much more in at that stage from the architecture.
Yeah, also always know the best way is starting in the lab. It's more easy for us, and then try to apply that knowledge on the field or to create the hypothesis that then we can test on the field in a more strategic way, like focus only in that conditions. Maria, a question for you. Does the project look at the nitrogen fixation ability apart from applied nitrogen efficiency? And also I can add, that's something the nitrogen efficiency, or we are considering to measure the specific reduction of nitrogen emission as complementing of carbon or translate to equivalence in carbon. I'm asking you because I know that many people from UK is focused in this topic.
Thank you, Alberto. No, for this project, for the scope of this project, and due to the nature of the call and the restrictions, we are not looking at carbon. That's number one. Not in this specific. It was not in the scope, and we are not doing it. So I want to clarify that. Then second, we are not interested in nitrogen fixation. That's not the scope of the project either. We are interested in looking at the impact of root exudates in the transformation, not just the NH4 to NO3, but all the cycle, like a little bit the cycle that Alberto just presented very nicely with all it's much more than NH4 to NO3 to NO2. There's many more different. And when you reconstruct the pathways, you can see many of the effects. I have seen more impact, and that's just very novel a bit, but this is something I'm willing to share a bit. We have also, because of the approach with the microbiome, we have of analysis of the 16S and the function, we have actually also seen an impact in the latest stage, in the oxidation toward gas, which is something, I don't know how far we can go with this in this project, but it's incredibly interesting that we can actually, the oxidates also act on that, which will decrease the emissions, which is even a more concerning loss of nitrogen. And obviously what we want to link with the nitrogen use efficiency, that's why we are going to combine what we see from the microbiome and the impact of exudates in the functions, nitrifying nitrogen cycling functions, with an actual intake of the plant of more nitrogen. Is this impact on the bacteria in the nitrifying communities reflecting in an increase in nitrogen in the grain, for instance? And we are also going to run, so we are going to measure that. We are going to use isotopes to trace to track, the input of the fertilizer. Where is it going? Is it staying in the soil? Is it being affected by the plant? Because at the end of the day, we are not measuring the degree of suppression in nitrifying communities just for fun. It's for two things: to decrease the losses and emissions to the environment, but also for the plant to buy time to use better, to get more of the nitrogen that goes for the fertilizer. So we are going to measure that in the grain and then in the soil with chemistry and isotope.
And also was very interesting because this morning I was reviewing some information about different products linked with nitrogen, and I found in different vademecum that there is like 6-7 different components of nitrogen that alone or mixed together people are using in different applications, in different fertilization programs. For us, it's interesting to understand if the microbiome, together with the roots, can be beneficial in some place for only one of that compounds or other compounds, or why in some case people is selecting all together. No, that's something that for me is in the way of this project. Well, it's time to end. I try to answer very quickly and combine two questions. One of the questions is, which other beneficial roles of bacteria are you able to predict? Not only bacteria, also fungi. And together with the other question that is pest pathogen, and which account for 30–40% of reduction of productivity, how do you relate soil health and microbiome to reduce this effect and increase grower income? Well, first of all, exactly, when you look at the microbiome, you can find any piece of DNA that come from different bacteria or fungi that are present in the soil. There are some beneficial species and also detrimental species. In this case, we are able to identify pathogens. And pathogens are done and not with identification of pathogens, level of pathogens, and very important, ecology. It means what is the number and what kind of beneficial microbes by control we identify together with the specific pathogens. We are able to run some specific tests to identify the risks of the disease and try to prevent that. It's true that when we are testing biostimulants or other projects like this, combining genetics, etc., we can find differences on that disease risk pressure. For example, we apply or run some project testing conventional with organic agriculture. And in this case, we identified that the abundance of the pathogens is higher in organic than conventional, but the disease risk is higher in conventional than in organic. Well, this is something that we can test and is involved for sure. If we are feeding the soil with nutrients and also applying crop protection product, we are playing with the life that are present in the soil and taking the knowledge and the control of how you can influence that actions to improve the yield and the sustainability is linked with the soil health definitions. In that case, the soil health definitions allow to promote the functionality in the soil. And to promote functionality is necessary to promote diversity in different ways. And this is not about only crop protection of fertilization, is a combine of all together. Yeah. Well, thank you very much. I know that maybe there is a question or a few questions more, but we have not more time. We passed like 15 minutes. And I appreciate a lot that you were interested in this webinar. We are here and we can share our contact details to answer your question and to be in touch, hopefully in person soon. Thank you very much. I appreciate, and thank you, Maria, thank you, Silvio, to participate in this webinar. Your support in research in microbiome and genetics in the plants is super important to go ahead in a Green Deal. Thank you very much.
Thank you, Alberto, for the opportunity to introduce the project, and obviously for your fantastic contribution with expertise and the technological platform. So that's gonna make a difference. And I hope in a year's time we have already some results to share and how far we have gone. Always a pleasure.
Perfect. And finally, as I like to mention that better soil, better food, better life. Thank you very much. See you soon.
Thank you. Bye-bye.
Bye-bye.